Hello everybody, welcome back to my channel and welcome to part 2 of Immunology for A-Level Biology, Edexcel A and B. As I said in my previous video, which I will link down below and in the description, I also have some other videos which I've produced for Immunology for varying exam boards, which have got useful exam technique help at the end as well. So feel free to go and watch all those videos and get as much knowledge and learning and engagement as you can. The content for today's session includes physical barriers to infection, non-specific responses, and types of immunity. So for Edexcel A, these are the specification points which we're going to be covering. 6.11 and 6.12 for topic 6, which is immunity, infection, and forensics. So feel free to pause the video and have a read through these if they're relevant for you. Then for Edexcel B, we're going to be covering points 4, 5, 6, and 7 from topic 6, which is microbiology and pathogens. And this is from the point 6.7, which is response to infection. So again, if this is relevant for you, feel free to pause the video and have a read through. Before we get into the video, make sure to comment down below, how do you feel about immunology in A-Level biology? I asked this in my previous video, so if you've seen that, perhaps you're feeling a little bit more confident than you were, but hopefully this video will help you build that confidence throughout the whole topic of immunology within A-Level biology. And then whilst you're there writing your comments, make sure to subscribe. It's quick, it's free, it helps me out, it helps the channel, and it helps support this great community of biology whizzes and students that we've been supporting. So first we're going to be talking about physical barriers, so the human body's physical barriers to infection. Some of the major routes that pathogens take to enter the body include cuts in the skin. They also include the digestive system via contaminated food or drinks. If you ate some moldy food or a gone off drink, that could be contaminated. That can affect and allow pathogens to enter your body. Then some other routes include the respiratory system, so you can inhale it through your nose, and also other mucosal surfaces, so the inside of your nose, your mouth, your genitals. So these are all major routes which the pathogens take to enter the body. Now we're going to talk about the physical barriers to infection. So of course, first, this may seem very obvious to some of you, but we have our skin. So skin actually has a hard protein on the outer layer, and this is called keratin, and this is very effective at preventing the entry of microbes. Obviously, as we discussed just in the slide before, if you do get cut or wounded, then this does enable entry, but the process of blood clotting actually helps to seal that wound, which reduces the number of microbes entering your body. Skin flora. So these are large numbers of harmless microbes on your skin. So they habitate there, they live there all the time, and they help to prevent colonization of other bacteria which may be pathogenic. They occur naturally, as I said, and they're very well adapted to your skin's environment, whereas other bacteria aren't so suited to these conditions, which are created by salty sweat and excreted chemicals such as urea and fatty chemicals. So the presence of this natural harmless bacteria helps to protect you from harmful pathogens. We've then got stomach acid. So if you unfortunately do take something in through your mouth that travels down your esophagus, also known as your food pipe, that is pathogenic, then the stomach actually produces hydrochloric acid, which is usually of a pH of 2, and this is able to kill the majority of pathogens that enter via this route. Then for those pesky pathogens which are able to evade that hydrochloric acid, we actually have flora inside of our gut or our intestines. So these are able to compete with the pathogens for nutrients and space, and it essentially limits the number of pathogens that are able to live and survive in the gut, which makes it harder for them to infect the body. So our final physical barrier to infection is the lysozyme enzyme. So mucosal surfaces such as your eyes, your mouth, your nose actually produce secretions, which we're very well aware of actually, such as tears, saliva, and mucus, and they actually contain an enzyme known as lysozyme, and lysozyme kills that bacteria by damaging their cell walls, and this causes bacterial lysis, which is essentially the bursting of those cells. Now we're going to talk about non-specific responses to infection, such as inflammation, interferon production, fever, and phagocytosis. There are a number of non-specific responses of the body to infection. So we've got inflammation at the site of infection, the production of interferons, fever, and phagocytosis. So we're going to cover all of those in a bit more detail now. First up, we've got inflammation at the site of the infection. So if you get an injury, a cut or a graze, any kind of wound which enables the microbes and other foreign material to enter the body, then a blood clot is able to rapidly seal that wound. But inflammation at the site, which is known as the inflammatory response, also helps to destroy those invading microbes. So while we talked about the physical barrier of the blood clot being able to seal that wound off and prevent the microbes entering the body, it's the inflammatory response which helps to destroy those invading microbes. This process involves the immune system cells recognizing the foreign antigens on the surface of the pathogen. So if you've seen my previous video on immunology, we talked about antigens and antibodies there, and we talked about how immune cells actually recognize those foreign antigens on the surface of pathogens, and this is what kicks off the immune response. So next, after they've been recognized, these damaged white blood cells and mast cells, which are found in the connective tissue just below the skin, they're able to release chemicals such as histamine, and this chemical triggers inflammation. Histamine causes vasodilation, which is the widening of blood vessels around that site of infection, and this increases the blood flow to it. The reason that this is important is that it essentially allows the white blood cells to rush to that site so that they can start destroying the microbes that have entered via that wound. Histamine also causes increased permeability of capillaries. So cells in the capillary walls separate slightly, so the vessels leak. So you may have heard of histamine before in terms of allergies. So some people will take antihistamines if they have allergies, for example, to, I don't know, grass or pollen, especially in the summertime. So this is a response that produces histamine when we don't want it to. Obviously, because you don't want to be crying when you smell pollen or something, because although it's foreign, it's not harmful. But that's an allergic response. But in this case, we obviously do want the histamine to be produced, as we want the inflammation to occur so that we can tackle and target those microbes. As I just said as well, it's that increased blood flow and permeability causing plasma fluid, white blood cells and antibodies to leak from the blood into the tissue, causing something known as edema, and that is purely swelling. Once that swelling has occurred, the intact white blood cells are able to start destroying the pathogen, and they do this in a number of ways. Our first way that antibodies actually destroy microorganisms or label them for destruction is known as agglutination. So microorganisms are clumped together for easier phagocytosis, basically. So as we talked about in our previous video, each antibody has two binding sites. So an antibody the two pathogens at the same time, which allows them to become clumped together. So then the phagocytes can bind to the antibodies and phagocytose a lot of pathogens all at once. So it speeds up the process of phagocytosis and destruction of microbes. We then have opsonization. So this is the process of pathogens being coated with antibodies, which marks them for phagocytosis. So when the antibodies bind to the antigens on pathogens, this actually can block the cell surface receptors that the pathogens need to bind to the host cells. So that means that the antigen can't attach to the host cells and hence they can't infect them. So this is a really clever process of preventing pathogens from infecting host cells. And it's kind of similar sort of like a competitive process, which is again similar to enzymes in the case of if one substrate takes up the active site of an enzyme, then another substrate can't basically bind to that enzyme, it can't be broken down. So it's a very similar process. Then we have precipitation, which is also known as neutralization. So soluble toxins are essentially made insoluble and inactive. So antibodies bind to toxins produced by pathogens, preventing them from affecting human cells, and so the toxins are neutralized, which is also known as inactivation. So then the toxin antibody complexes are also phagocytosed, so that that toxin that's present with the antibody can't cause any more destruction. Then we have lysis, which is also known as bursting, and this, as you can see from the image, is the breaking open of bacterial cells. So antibodies activate the complement system to destroy bacterial cells by lysis, which is punching holes in that cell wall, causing it to break open, that bursting, and then iron and water flow into the infected cells, which helps it to undergo lysis, so it swells and bursts. That was inflammation. We've then got the production of interferons. So this is a chemical release from infected cells which prevents viral replication. So when cells are infected with viruses, they produce these proteins called interferons, and the interferons actually help to prevent viruses spreading to uninfected cells. The way that interferons prevent this viral replication is by inhibiting viral protein synthesis. So they activate cells involved in a specific immune response to kill infected cells, and they also activate other mechanisms of non-specific response, such as that promotion of inflammation to bring immune cells to the site of infection. So that was interferon production. Now we're moving on to fever. So the hypothalamus, which is in the brain, sets the body temperature naturally higher. So this decreases the speed of pathogen reproduction and increases the rate of the specific immune response. So by increasing that temperature, this means that the immune cells can actually work better, whereas the pathogens can't reproduce as quick. So a lot of the time people think that it's the pathogen making you feel that higher temperature, feel that fever, but it's actually the response of the body in order to protect you, to help increase that immune response and to prevent the rapid reproduction of pathogens. That was fever, which I do think is a really interesting one, because I think a lot of people think that it's the pathogen making you feel that, when actually it's the body trying to help you. But now we're going to move on to phagocytosis. So the first stage of phagocytosis is the damaged cells and pathogens releasing chemicals which attract the phagocytes to the site of infection. And then that phagocyte is able to recognize the antigen on the pathogen, and the cytoplasm surrounds that pathogen, so it forms an invagination. And then the neutrophils are actually the phagocytes which arrive first, and they're able to engulf about 5 to 20 bacteria, but they then become inactive very quickly and die. Whereas we have macrophages, another type of phagocyte. Again, if you see my previous video on immunology, we talk a lot more about that. But these are larger and longer lived phagocytes, so they're able to destroy as many as 100 bacteria. So a lot more than neutrophils can, and they ingest debris from damaged cells and foreign matter. The next stage is the pathogen and ingested material being enclosed in the cell in a vacuole, creating a phagosome, which is a phagocytic vacuole. So then within the phagocyte there are lysosomes which contain lysozymes, which you talked about previously. So then these lysozymes are hydrolytic enzymes which are able to hydrolyze those pathogens and break them down. Lysosome fuses with the phagosome, which exposes that pathogen to the lysozyme. So that means the lysozyme is able to hydrolyze the pathogen, and any soluble useful molecules are absorbed back into the phagocyte's cytoplasm. Finally, the phagocytes present the antigen of the digested pathogen on their surface and become an antigen-presenting cell. So this is essentially a summary of phagocytosis and the non-specific defense system. So you can see here we've got the bacterium, the pathogen with the antigen on, being picked up by the receptors of the macrophage. We've then got phagocytosis occurring here with the formation of the phagosome surrounding the bacterium. And then we've got the phagolysosome, when the phagosome and the lysosome fuse, and then you've got the bacterium being digested by that lysozyme enzyme. And then finally, the phagocyte becomes an antigen-presenting cell. Now we're going to talk about types of immunity. So we're going to talk about primary and secondary immune responses, immunity types, and then we're also going to talk about vaccinations, which links to that immunity. So the primary immune response. So this is your very first exposure to that pathogen. So the first time a B cell comes across a non-self antigen that is complementary to its cell surface receptors, the production of sufficient antibody producing cells takes about 10 to 17 days. days. So the reason that you suffer symptoms is because of the time taken to produce these antibodies. The secondary immune response, so this is when you're reinfected by the same pathogen, so you're exposed a second time around. So the B memory cells are immediately able to differentiate to produce plasma cells which release antibodies. So this means that the pathogen is destroyed before the symptoms are produced, hence you don't get ill. This is known as natural active immunity. So when you're first exposed, you can see here on the graph from the time, so when you're first exposed, the concentration of antibodies is very low, and you have your primary immune response, and although it produces antibodies, it's quite a low concentration, and hence it takes a longer amount of time to tackle. Whereas when you're exposed a second time around, the immune system is able to produce a high concentration of antibodies very, very quickly, which helps you to prevent yourself being ill. That was natural active immunity, and we're going to talk a bit more in detail about the other types of immunity as well. So as we said in the previous slide, we've got natural active immunity. So natural means the normal body response, and active is that body cells are involved. So this is the normal response to a foreign antigen. We then got natural passive immunity. So this is the protection developed in another individual, and then natural again being the normal body response. So for example, antibodies cross the placenta to protect a baby whilst its immune system develops, and then when it's born, that breast milk, especially the ingredient colostrum, also contains antibodies which is given to the baby from the mother. We've then got active artificial, so artificial being a medical procedure. So vaccines, for example, use antigens from pathogens in a harmless form to cause that primary response, and then reinfection therefore allows for a secondary response. And then finally, passive artificial. So for example, an antiserum contains antibodies, usually from the blood of animals. Following an exposure to an antigen, you're able to be injected with these antibodies, which neutralizes the antigen, but doesn't actually provide long-term protection. This is the same as the previous slide, just set out in a different way. I know different people learn in different ways, so I thought I'd include this information written in a slightly different style, slightly different display. So if you want to use these for your revision instead and have a read through of this, then you can absolutely do that as well. Okay, we're now going to talk about vaccines. So whilst your B cells are dividing to build up the numbers to deal with a pathogen, so during the primary response, this is when you suffer, which we talked about previously, you suffer symptoms. So a vaccination is able to help you avoid this, and vaccines contain antigens that stimulate that primary immune response against a particular pathogen without causing disease. So this results in your body producing those memory cells, and means that if you are infected, you're able to respond to that immediately, but you don't have to suffer symptoms of the disease at any point. So some vaccines actually contain many different antigens, and this is in order to protect against different strains of pathogens which are created by a process called antigenic variation. Now we're going to talk about the advantages of vaccinations. So the first is herd immunity, and if you don't know what that is, don't worry, we're going to talk about that a little bit more in a second. We've then got the fact that when more people are immunized, the number of cases of the disease decreases, and this is because usually if you're immunized, you're much less likely to catch that disease, and hence the number of cases decreases. And then in comparison, the chance of falling seriously ill or dying from a disease is usually far greater than the chance of experiencing a serious side effect from a vaccine. So for example, unfortunately, some people did die from blood clots from a COVID-19 vaccine. However, the number of people that died from the side effect of the vaccine compared to the number of unvaccinated people who caught the disease, unfortunately, and did pass away, is vastly smaller. So obviously, we've had hundreds of thousands of people all over the world who have sadly lost their lives due to COVID, whereas only a very small percentage have due to the vaccine. And those that are vaccinated have had much greater chance of surviving the disease if they have caught it. Also, another advantage is that using a vaccine is usually much cheaper than treating a very ill person. One vaccine for one person, or two or three, depending on the disease, is much cheaper. They can be produced at a high rate, whereas if you're treating a very ill person, you may have to use a number of different therapies, which can get very expensive. There may not be the right therapy out there, which results in the need for research and more resourcing. So that's why basically vaccines are usually cheaper than treating an ill person. Herd immunity. So enough people basically have to be vaccinated to make transmission of a disease very unlikely. And it usually requires like 80 to 90% vaccination. And this is what herd immunity is. So immunization and vaccination are usually used interchangeably, but they're actually two different key terms that you need to know. So immunization is the process of protecting people from infection with passive or active artificial immunity, whereas vaccination is the process by which this is achieved through the use of attenuated antigens. So attenuated vaccine, or a live attenuated vaccine that is sometimes referred to as, is a vaccine created by reducing the virulence of a pathogen, so reducing its harmfulness, but still keeping it viable or live. So this means attenuation essentially takes an infectious agent and alters it so that it becomes harmless or less virulent to you, so hence you don't catch the disease when you're vaccinated, or you don't become ill from it. So this is basically the process of herd immunity. It's making sure that lots of people are vaccinated, usually 80 to 90%, so that transmission of disease is very unlikely. Okay, as I talked about previously, unfortunately, there are some risks with vaccinations, so there can be side effects or reactions. Usually these are very minor, although sometimes they can be fatal, but as I put there, this can be rare. So I've given that example of the blood clots with the COVID-19 vaccines. There was a time when there was speculation that the MMR vaccine, so the mumps, measles and rubella vaccine, caused autism. So lots of parents didn't get their children vaccinated, which meant that these diseases spread easier and the cases rose. But the World Health Organization have proved that there is no link between MMR vaccine and autism. So unfortunately, this was a bit of speculation that got out of hand and caused disease to rise. So now we're going to talk through some implications of vaccinations. So ethical implications. So for example, money spent on vaccines could instead be spent on education or preventing famine. Obviously, we're lucky in this country and in the Western world and in more developed countries that we're able, that we have good education systems in place, and preventing famine isn't something that we have to think about because we have those food and those resources. We're very lucky to have that. Whereas in some countries, less developed countries, poorer countries, they really have to prioritize specific things and work out what's most important. And what they should spend their money on. So unfortunately, sometimes this money has to go elsewhere rather than being spent on vaccines. There's also social implications. So vaccines do need to become accepted. Obviously, there's people who are anti-vaxxers, or people who have come up with conspiracy theories, who think that vaccines aren't good, or that they've been put out there by the government to inject us with all sorts of ridiculous things. So it is really important that these vaccines become accepted, so that more people get vaccinated, so that 80–90% of a population are vaccinated, so that herd immunity can occur. There's also economic implications. So the treatment, control and prevention of endemic diseases can be very expensive, and obviously we've not only experienced an endemic disease but a pandemic disease, which is obviously be very expensive worldwide. So there's quite a few implications when it comes to vaccinations. So just to quickly finish off then, I found a couple of interesting articles about opting out of vaccinations and how it puts the greater population at risk. Essentially, so these are just two that I found very interesting, that if you may want to have a read for a bit of wider reading for your A-level. So we've come to the end of our second part of our immunology videos for Edexcel A-level A and B. So make sure to comment down below how you now feel about immunology after watching this video. You can use these emojis along the bottom if you would like to, or you can write out a longer comment if you have some more to say and you have some more feedback. I'd really appreciate it. thank you so much for watching, and don't forget to subscribe down below. It's quick, it's easy, it's free. You can help me out, help the channel, and join a really fantastic community. And if you do have any more questions, feel free to comment down below or message me on Instagram at Biology with Gracie. And I will see you in my next video. Bye guys.